Thank you, and thank you all for coming. I'm Steve Linderman. I'm a clinician, scientist, and the CEO for Absco Therapeutics. Like Andy was just mentioning, we're really focused on how we can deliver drugs more effectively directly into a tumor to create immune education that can fight off that tumor at the source and also go through the body and fight distant sites. We are very excited to be starting with early cervical cancers in gynecologic oncology. And so, towards that end, just to plant the seed, we are looking for some support with women's health oriented business advisors. We have a very rapid path into clinic. In about 15 months, we're submitting our IND. And so, looking for some support with clinical trial ops and regulatory. Capital efficient path, thankfully we just got an NSF grant and a term sheet for our seed, so we're looking for investors that are aligned to help us fill out that syndicate. And we've been really actively building out our platform as well, so looking for some connections for potential strategic partnership. And with that, to really orient you to the intratumoral space, it's a fascinating concept. So directly inject a drug into a tumor with the idea that you can directly modify that tumor microenvironment and create immune education at the source without needing your drug to go systemically through the body and cause toxicity. Historically, however, this has really struggled to gain traction in the clinic. And there are a few big reasons for it. The biggest one is a study out of MD Anderson a few years ago showed only 1 in 4 patients actually had drug retention in the tumor an hour after injection. So horrible on-target hit and delivery, because when you inject, the drug just leaks out if it's a liquid. In addition, you can't actually see it on imaging. So you can see a needle tip, but you can't see the drug itself. And when it tracks along the needle, it means that you've missed. That means that you end up needing really frequent repeat injections. And so we paired up with Bob Langer and Gio Traverso's lab to build this technology where we use a thermogelling polymer. We can deliver it directly into the tumor. It gets retained on site. We can track it on imaging in real time, either on CT or ultrasound. And then we get controlled release of a variety of different immune stimulating agents. Our lead asset leverages a small molecule, imiquimod. So to show that this actually works, this is a pig study that we did several months ago. We can inject it in many different places throughout the body. In each case, it is very easy to see. It stays localized, it doesn't leak, no toxicity issues. This is a mouse-based PK study where we showed that we get about 1,000x higher drug loads in the tumor compared to the serum. So really taking out that concern about systemic toxicity because the drug stays where we put it. And we get it there for an extended period. This was at 5 days, we're mapping it out. It should be about 7 to 10 day release, intentionally to be able to get immune education. And then when we think about efficacy, this is one of the really cool parts, that we can put a tumor on both sides of the animal. When all you do is give systemic checkpoint inhibition, they all die because they're immunotherapy resistant. But when we inject our therapy into only one of those 2 tumors, both of them in the responding animals will fully shrink, go away, no tumor regrowth out to 90 days, no tumor regrowth on rechallenge. And so where are we going with this? The topical version of imiquimod is already approved as a cream, used mostly for basal cell carcinoma. It's also used off-label for cervical cancers. It works really well in that it gets histologic regression. It doesn't work well in that the cream isn't formulated right for mucosal use, so it burns the vaginal mucosa. It's uncomfortable for patients. It's very difficult for them to apply it and actually get it to the cervix itself. But we think that this de-risks the clinical path forward and means that we can do a direct inject into the cervix with a monotherapy, get safety and efficacy data. It's a very straightforward study. It's a market in and of itself, about 180,000 patients in the US annually with CIN23, which is the precancerous version of cervical lesions. The current treatments are surgical, meaning they remove the tissue. We instead can avoid that need. So do a direct injection, preserve the cervix, and allow the woman to actually continue to have children after that. We've been getting great enthusiasm from gynecologists, from patients. Like I was mentioning earlier, we're looking for some women's health business advising support as we move in this direction. And we think this is a launch pad. So move from early cervical into invasive and other advanced cervical cancers, other HPV-driven tumors, other virally driven tumors throughout the body, and long-term other deep tumors like metastatic colorectal and breast cancers. These are large markets, looking at about a 1.8 billion start, multi-billion follow-on indications. We've been thankfully gathering a ton of traction over the last month even. Just submitted an FDA Interact at the end of March, got an NSF grant for an SBIR that was recently started and awarded. Got a great score on an NIH SBIR that should get awarded soon. and just got a term sheet for a $9 million seed round to be able to carry this forward. So we have 3 million committed, 3 million soft circled, and we're trying to get that closed over the next few months. We've been building out the advisory team on the Gyn-Onc side, on the business side, on the non-clinical side, and we're really excited to continue building this momentum so that we can drive this technology forward towards clinic. The team, in addition to myself, we have Dave Altrider, an expert in CMC. He's been in several Langer Lab companies actually previously for about the last 25 years, who's brought over a dozen different technologies through the manufacturing parts in the clinic. Cheryl Elkins, former Taurus CTO and then Chief Compliance Officer, has a lot of regulatory and compliance background, so she's advising us on that side. Gil Covarrubias came out of MIT's Paula Hammond Lab to be able to really drive our cancer immunology forward. On the advising side, Jennifer Scalisi runs Gyn-Onc at Emory University, which is a very concentrated area with a lot of cervical cancer patients. So she runs a cervical cancer clinical trial consortium. Jeff Feiner is out of Third Rock Ventures in Septerna. He's been a long-term mentor of mine since I was at Third Rock. Shashi Ramaya is actually one of the catalysts here in the room who's been supporting us with non-clinical talks. So where do we actually see this going? This is a long-term projection slide, but we're about a year out at this point from IND to move into early precancerous cervical lesions, and then we can see that expanding into other cervical cancers. And so towards that end, we're looking for a women's health business advisor to help us drive this forward. Clinical trial ops to help us once we actually get into the clinic. We have a very MD-heavy team and advisory team, but looking for people that have more of the clinical trial executional experience. With the fundraising side of things, we're building out the syndicate currently, and then working based on grants to connect in with strategic partners. We've been showing that we can deliver a variety of cargoes like nucleic acids, and so we think that opens up some pretty interesting opportunities. And so with that, I'll leave you today, but I would love to talk to you more at the table outside, share a little bit more flavor of what we're doing, and thank you so much for your time.